Hello everyone, my name is Wolfgang Hankel and I'm here at the GSC14 and I'm sitting here with Alexei Drummond from New Zealand. And I'm going to ask you questions about the GSC. First of all, may you introduce yourself and tell us about your fields of research.
So I come from the University of Auckland in the computer science department, but my background is in evolutionary biology as well as computer science. And my field of expertise is really in Bayesian statistical analysis of genetic data, so building phylogenetic trees. But so the sequence analysis side is where my expertise is, but as we'll talk about later, it's the collision between ecology and genomics that's really what's getting these genomic observatories going and where I'm interested in.
Sounds interesting. Is it the first time that you attend the GSC meeting?
This is the first time that I've attended the GSC, and it's interesting for me because we've been increasingly dealing with lots of sequence data in my lab as we start connecting with more biology groups outside of our department and doing our own field research and sampling, but we haven't really got involved in the standards until quite recently.
So what were the themes that were particularly interesting to you?
Well, the main thing was the special focus on genomic observatories, because we have our own GO that we've just started up in New Zealand a couple of years ago. But I mean, more generally, this kind of next-gen sequencing data that we're starting to generate for other projects as well really starts to require a lot of standards.
What, from your point of view, are the main contributions from the GSC to a wider community?
Well, I mean, I think, you know, getting scientists to coordinate their efforts is clearly important globally for the scientific community to best use our data, as if we can all talk in the same language and best combine our efforts into sort of more broad meta-analyses that cover the whole globe. So that's very important, and I think, you know, it's not always easy that scientists aren't necessarily doing these things themselves because the motivations don't always line up, and so you need some sort of coordinating body like the GSC, I think.
That makes sense. So now coming to the GOES initiative, what is a genomic observatory from your point of view?
Well, I mean, for us, the concept of a genomic observatory is an intensively studied location where the primary tools that we're using are genomic tools. And that's not necessarily because we're interested in genomes per se, but they are very easy, accurate and repeatable markers of ecological communities and activity in ecological communities.
Can you briefly describe the work that you do at your genomic observatory?
So our observatory is a offshore island focused around an offshore island called Hauturu and the Hauraki Gulf. It's 30 square kilometers, and it's a heavily forested island. It's sort of pristine. In some sense, it's a key importance for New Zealand's biodiversity. It's a conservation focus. And it's a national park. And what's interesting about it is it's sort of the last remnants of untouched terrestrial forest. Not the last remnants, but some of the least modified. And so it's really where we're focused on in terms of understanding what New Zealand might have looked like before humans arrived 800 years ago. And when we're thinking about conservation and restoration in other parts of the country, Hauturu is our model for what things might look like.
Why would you say is setting up the GOES network important?
Well, any intensive research that really spans, so in our case we're looking not only at the microbes in the soil, but the invertebrates in the soil, and all the way up to the plants and the animals living on the island, genetically sort of characterizing the entire ecosystem. And a lot of different technologies come into play, a lot of different problems with data analysis and data storage and management. And I think being able to gather information and expertise from other similar sites with similar problems is a key for us to not reinventing the wheel and to using best practices.
From your point of view, what would you say will the GOES infrastructure tell us that the infrastructure that is out there cannot tell us yet?
Well, I mean, you've heard, you know, there are efforts to, and a real focus on providing contextual information and metadata along with our sequence data. But there's different levels of that, and when you're intensively studying a single site, and when you're really doing experiments where you right alongside the collection of environmental sequences, you're running temperature and humidity monitors, and you're running in a dense array, and you're doing soil chemistry analyses, you really know a lot more about how that data fits together than what you might with some of these other approaches. And so it requires a sort of another level up of integration and understanding of how the data connects.
Talking about integration, how would you say, or what is the best way to integrate the GOES into already existing infrastructure? So,
I mean, I think that's a key thing, and that's probably why we're here. For us, already in the first few talks, we've been seeing the kinds of frameworks and checklists and various software tools that are being developed, and we're really keen on using and plugging into as much of those as we can. I mean, one of the problems is a real proliferation of different options for how you want to manage your data, and so to make good decisions so we're not developing a huge amount of infrastructure within our own group that would repeat other people's efforts is why we're here, really.
Good. So thanks a lot for your time and answering the questions, and have a good meeting.
Thank you very much.